Today's webinar is sponsored by Intilaris Life Sciences. Intilaris is a Swiss-based clinical development consulting company and is extremely pleased to be able to sponsor this presentation today by Ken Getz. So as I was saying, today's webinar is new insights on optimizing protocol design practice to maximize development performance, sponsored by Intilaris Life Sciences. And with that, I'd like to turn it over to Torsten Friedel from Intilaris to say a few words about Intilaris. Torsten?
Thank you. Thank you, Robert. Torsten Friedel, Intilaris Life Science. Quick introduction to Intilaris Life Sciences. We focus on productivity improvements in clinical development organizations. So basically, we help to improve the productivity and optimize processes in clinical development, phase 1 to phase 3 trials. In that area, we are providing consulting services and also technology, and we help our clients to optimize study design process, protocol development process, and basically increase productivity in clinical development. Thank you very much. I'm handing back to Robert.
Okay, thank you, Torsten. And just for everyone's notice, we will be recording today's session, and we will be able to make that available afterwards on the Intilaris website as well. And with that, let me now turn it over to Ken Getz. And so Ken is the head of the Tufts Center for the Study of Drug Development, CSDD, and is an internationally recognized expert in R&D related to clinical trial management practices. And Ken's research has expanded over 20 years of original research related to benchmarking R&D management practices, and his efforts have contributed to industry-wide understanding of these critical markets and to the improvements in management strategy and execution. Ken's research has covered a broad variety of topics related to protocol design complexity and clinical research efficiency and effectiveness, and his work is considered by many to be pioneering efforts. And with that, I want to say we are extremely pleased to have Ken join us today. And with that, Ken, let me turn over the control to you.
Thank you so much, Robert, for that introduction, and I want to thank Intilaris for hosting this webinar today. It's really my pleasure to be here and to give this talk and to share these insights with you. And I want to thank all of you for joining the call today. It's a privilege for me to share with you the highlights and insights from the work that we do at the Tufts Center. And there's no shortage of insights that come from our work, new questions that we can evaluate and try to understand. And so I welcome your comments, your thoughts, some of the insights that our research results may generate for you. I want to point to a lot of the places where you can go to gather a lot more information behind many of the results that I'm going to share with you today. As Robert mentioned, we're coming up on roughly 17 years of research specifically focusing on protocol design practice and opportunities to optimize design. And these are just some of the major publications that contain detailed descriptions and data. We have another paper coming out next month in Nature Review, which will provide a lot of updated benchmark data. So I encourage you to keep your eyes out for a lot of our publications if you're interested in more details and more information. I want to mention, before we dive into the specific insights and the results, just the basic methodology that we use for all of our studies. And this is very, very important because all of the data is coming from actual protocols that have been provided by the companies themselves. Every single study that we've conducted in this area has used what we call a working group model, where we have anywhere from 16 to upwards of as many as 30 companies small, mid-sized, large pharma and biotech companies that are sharing their protocols with us. And we typically have the protocol authors or individuals from each of the companies code their protocol design behaviors and practices and code many of the variables. So we don't make up any of that coding. All of this is done by the people who are the closest to the protocols themselves. So what I'd like to do today is really touch on 4 key areas that all help tell the story about opportunities to optimize protocol design. I'm going to begin really where the presentation should end, and this is a little unorthodox, but it's critical to start with the key takeaways because that helps change our philosophy about where we should focus on opportunities to optimize design. I'll then talk about specific protocol design practices, and I'll characterize those practices for you, and then we'll relate those practices to impact. Many of the design decisions that we make have a direct impact on cycle time and cost and quality of our development programs. And so we'll look at some of the data that really helps us understand impact. And then I'll close with a discussion, high-level discussion of some of the major opportunities that exist to optimize protocol design. So to begin with the key takeaway concepts, I want to really offer to you the common questions that we hear when we give presentations and when we facilitate discussions based on the results of our studies. Are you saying that protocol complexity is bad? Should we look at our regulatory agencies as the primary party to blame? What specific procedures can we remove to make our protocols less complex? Why do you believe that clinical trials are a waste of money? And I'll talk more background about that question in a minute. And lastly, protocol amendments are typically implemented to improve design. Why do we recommend avoiding them? So let's start at the top. We are not suggesting that protocol complexity is bad. What we're suggesting is that protocol complexity continues to rise and we must find ways to manage it more effectively. And we offer, through insights in the research, ways that we can really target practices and solutions to support a better management approach. Are regulatory agencies to blame? I think you'll see in a moment that much of the blame rests with the specific decisions that we make within pharma and biotech that will ultimately trigger the endpoints and procedures to support them. What specific procedures should we remove? That's nearly impossible to answer because it's entirely specific to each individual protocol, and that's part of the important message here as well, that there isn't a way to generalize all optimization activity, but instead to suggest strategies that have to be tied to each individual protocol and the relevance of that strategy to optimizing that specific research effort's needs. Why do we believe that clinical trials are a waste of money? That comes directly from a journalist who heard me talking about an optimization approach, a way to essentially target non-essential procedures and to try to remove them to improve protocol execution feasibility. And those specific procedures have a very, very quantifiable monetary value. And so these are some of the optimization opportunities, and they have a direct impact on study budgets. And lastly, I just propose to you, and many of you know this already, that protocol amendments we typically view as a problem, but it's perhaps better and more correct to view the amendment as a solution to an underlying problem. And that's what we're trying to get at here. This is very recent data that will be published shortly, just characterizing the rapid growth in complexity across a wide variety of measures. And here we're looking at averages within 2 observation periods for the typical phase 3 pivotal trial protocol, separated by a 10-year time horizon. Total number of endpoints has nearly doubled, and I'll show you more about those endpoints in a minute. Total number of eligibility criteria has increased by 60%. The total number of procedures has grown dramatically for the typical phase 3 pivotal trial protocol across all therapeutic areas. Number of procedures per visit has increased as well, because we have typically not seen the duration of our studies increase to a large extent. And as a result, each visit, we are conducting more procedures for each study volunteer, on each study volunteer. The proportion of procedures that collect data that is not tied to a primary or key secondary endpoint has now approached a third, up from less than 20% in that earlier time period. And just one last data point for you, we're approaching the collection of nearly 1 million data points for a single phase 3 pivotal trial. And this is all data that has to be cleaned, it has to be managed, curated, archived, stored, analyzed. So that just another measure of the incredible complexity and volume of data that has to be managed.